Yeah, we're now recording.
Brilliant. Well, good morning, everybody. Welcome to a presentation as part of your Marrow National Conference. I'm really excited to be here. I've always wanted to attend one of your Marrow conferences. So I'm very happy to be here. And I'm just going to talk to you a little bit today about the Anthony Nolan Research Institute, although my slides seem to have stopped moving forward.
Could you, if you try clicking the arrow buttons at the bottom, in the bottom left, does that help? On the actual presentation.
Okay. Yeah, that works. Thank you very much.
Yeah, okay.
So sorry about that. I'm just going to talk to you a little bit today about some of the work that we do in our research institute, in particular our patient donor project, which is one of Anthony Nolan's longest running research studies. And my name's Nima. If you have any questions at all, please feel free to put them in the question box, and we'll answer those at the end if that's okay. Also, if there's any technical issues, if you can't hear me, please just pop in a message and someone will let me know that you can't hear me, and we'll rectify that as soon as we can. So I'm going to be a little bit rogue, and I'm actually going to start with my acknowledgements. The Anthony Nolan Patient Donor Project and a lot of the research that we do at Anthony Nolan couldn't happen without not only the support of obviously the patients and donors who take part in our studies, but also to everybody, including all of you who raise money for Anthony Nolan, who raise awareness about Anthony Nolan, and who help recruit donors onto our register. So I really want to start by saying a very big thank you to all of you for the work that you do to help us, and we wouldn't be able to do some of this research without your help. So a very big thank you to all of you. I'd also like to just acknowledge all of the UK transplant centres who support this research, and lots of my colleagues, both at Anthony Nolan and at other organisations, who are really a vital part of the work that I'm going to talk about today. So there are many factors that affect how well somebody will perform after a transplant. And they include a combination of genetic factors, such as the HLA genes. And the HLA genes are those genes that we talk about when we are considering tissue typing and matching patients and donors. And I'll talk a little bit about HLA as we go along. It's also dependent on non-HLA genetics, and we're really just beginning to delve into that world of trying to understand what role other genes than the tissue typing genes have in transplant outcome. And also a lot of clinical factors, including information about the patient, the kind of disease that they have, and what stage of their disease that they're at the point of transplant. But essentially, all of these factors have to come together in order to get a successful outcome. And the work that we're doing is actually trying to understand how all of these individual factors play a role together, and how possibly we can utilize some of this information to not only find the best donor, but to find the best donor for a patient who doesn't have the best donor. So the Anthony Nolan Patient Donor Project has been running for over 20 years, and we aim to determine how genetic and clinical factors impact on the patient outcome. And we collaborate with every transplant centre in the UK that performs unrelated donor transplants. So there's over 30 different centres at the moment that we are collaborating with at any one time to collect samples and collect data. This is the Patient Donor Project team. There's a number of people involved in this, including our medical director and a fantastic group of young scientists who are really making great strides in the work that we're trying to do, and who are taking care of the project on a day-to-day basis. We aim to recruit every patient in the UK that receives an unrelated donor from the UK aligned register. And we also recruit the donor themselves. And what we do in terms of recruiting these patients is we collect a blood sample from them, and for the patient that has to be before they've had their transplant, because we want the blood sample to be of the patient's original cellular material. Obviously, after the transplant, the cells that will be in their bloodstream will be of donor origin, and we want to understand the original genetics of the patient. We also need to know what happens to the patient after their transplant. So we need to have clinical follow-up data on these individuals. And we do that by collaborating with the British Society of Blood and Marrow Transplantation and Cellular Therapy, who are the UK register for all transplant outcome data. So when we receive the blood sample, there's two different processes that we do. The first thing that we do is we actually extract DNA from some of the blood samples so that we can do the genetic analysis. And for anybody who hasn't seen DNA before or extracted DNA, this sort of white cloudy little lump here in the bottom of this tube is what DNA looks like once you've first extracted it from the cell that you're interested in and before you actually have finished preparing it for use in your downstream applications. The second thing that we do is we take a whole blood sample, just like this blood here, and we actually take out of that blood, we extract the white blood cells. And we do that by layering it on a concentrated sugar solution, which is this clear solution at the bottom just here. And when we spin that in a centrifuge and subject it to really strong forces, what we are able to extract, and if you can just see this sort of cloudy layer here, we are actually able to separate out the white blood cells from the remainder of the cells and the other material that's in the blood sample. And so we can harvest that for downstream applications. So before I go on to a little bit of the numbers around our patient donor project, I thought it would be important just to touch on what impact COVID has had on our patient donor project. And actually, for the first time in the history of the patient donor project, we actually had to stop recruiting patients for the period of the immediate lockdown. So that was between the 25th of March and the 10th of July. And for the first time again, our Anthony Nolan Research Institute was actually closed. It's never been closed before, other than sort of public holidays. But we came back in July, we're back up and running and things are as busy as ever, and we're back recruiting to the patient donor project. So this work is continuing after a little hiccup, and we're very hopeful that going forward, we may have a few more challenges ahead, but we should still continue to recruit to the study. So as you can see here, this is just looking at the number of eligible transplants with the Patient Donor Project over the last 24 years or so. And what you can see is that mirroring what's really happened in the transplant world, there's been a steady increase in the number of eligible transplants that we're able to put onto the study. And certainly around about 2012, when we started recruiting donors from the aligned UK register as well, we've seen a massive increase in the number of transplants that we're able to recruit to the study, which is brilliant because we often need huge numbers of patients and donors in these studies to be able to have the statistical power to do some of the work that we want to do. One of the challenges that we do face is that while we collect almost all of the donor blood samples that we want to collect, all of the donors that we want to recruit into the study, unfortunately we don't always get the patient blood sample as well, and we actually need both to take a pair forward for further analysis. And this is often because the patient is not able to give a blood sample at the time of transplant, or often patients are going through enough, they don't really want to have to consider taking additional blood samples, and we completely understand that. This is a completely voluntary study for people to take part in, but it does affect the number of individuals that we do have in the study at any one time, and that's a challenge that we're always working to try and improve. So I mentioned the HLA genes and how they impact on transplant outcome. And this is just a cartoon diagram here of the HLA molecules. As I said, the HLA genes encode the molecules that make our tissue type. And there's two different kinds of molecule that we have that are known as the class 1 and the class 2 molecules. The class 1 molecule is encoded by a single gene. And the class 2 molecule over here is encoded by two different genes. So one encodes the pink part of the molecule and the other one the blue part. But essentially, even though they are structurally a little bit different, what these molecules do is present pieces of material, we refer to these as peptides. It presents this information to our immune system. And the HLA molecules themselves are responsible for our immune system recognizing what is us and what isn't us. So recognizing self from non-self. And that's why they're so important in transplantation, and why when we're trying to match patients and donors, we try to match them as well as we possibly can. Because what we don't want is the molecules that we're putting in from the donor cells to look at patient material and see that as something that is non-self, because what they will do is start to mount an immune response against the patient tissue. So we're trying our best always to try and have the best possible match to avoid that non-self immune response that can occur. And there are 6 HLA genes that we're currently considering in matching patients and donors, and they are 3 in the class 1 family and 3 in the class 2 as well. So if we have a look at how this works in practice, and this is a very simplified way of looking at matching. Here we have Dad, and he has 2 copies of his HLA genes, one from his mum, one from his dad. And we're just looking at the class 1 genes, which are HLA A, B, and C. And you can see he's inherited 1 copy here and 1 copy here, and we refer to each copy of the genes that an individual has as a haplotype. Homer meets Marge, and Marge has 2 different haplotypes, so 2 different combinations of HLA genes in comparison to Homer. And they start to have kids. And so the first child comes along, young Bart here, and he's inherited this particular haplotype from his dad and this particular haplotype from his mum. Lisa comes along and she's inherited the same copy from her dad, but a different copy from her mum. And so on and so on. And I know that these 2 are not Simpsons, but it's really hard to find a famous family with 5 kids in it, other than the Kardashians, but I wasn't going to go there. So worst case scenario, Bart needs to have a transplant. The first thing that we will do is look through the family, and we find that there is, here Martin has the same combination of genes, he's inherited the same copy from both parents, and so he would be, in this case, a suitable match. But if Martin didn't exist, that's where Anthony Nolan comes in, and that's where our panel of unrelated donors, our volunteer unrelated donors, and our umbilical cord blood bank comes in handy, because we're able to look for a possible match where there wasn't a match within the family. So why are we looking for matches? So as I said, it's very important to find an HLA match because we're looking to try and stop that recognition of self and non-self by the HLA genes. But what we also know is that the better matched a patient and donor are, the better their outcome is. So here, this graph is just looking at the overall survival of individuals stratified by what matched donor that they had. So when we talk about an individual who is matched for both copies of 5 of the HLA genes, we refer to a 10 out of 10 match. If there's a single mismatch there, we refer to that as a 9 out of 10 match, and so on. And what we can see here is that this is data that's come out of the Patient Donor Project, but it's also mirrored in other studies around the world. And we can see that those individuals who have the best possible match, so a 10 out of 10 match, their survival is significantly higher than those who have a 9 out of 10 match. And then those with multiple mismatches, so an 8 out of 10 match or less, they do considerably worse. So we know that matching is really important in terms of patient survival, and we're always trying to find a 10 out of 10 match. This is really complicated though, because the HLA genes are hyperpolymorphic, which means that there are huge numbers, there are thousands of different tissue types that we need to be able to look for and match for during the process. So this is just a graph showing how the number of tissue types have increased over the last 30 years plus. And what we're actually seeing is that we have over 20,000 variants of just the class 1 genes alone. And we need to be able to characterize most of those, or ideally all of them, for our patients and donors. So this is a very complicated process for us. To give you a bit of an example of how this has affected us in the laboratory, traditionally when we used to do tissue typing, we actually used to use a technique called serology. And we would take the white blood cells of patients or donors when we were going to do the typing. We would use a panel of antibodies that were specific to the different tissue types that we knew about at that point. And we would stain the cells and we would see which ones gave positive reactions and which ones gave negative reactions, and we were able to determine what the tissue type of these individuals were. Predominantly now, typing is performed using DNA-based methods, but we do still use serology. It has its uses even in today's world with all of the different tissue types that we have identified. So to give you an idea how DNA technology has actually helped and hindered us a little bit, using serology, we were able to type somebody as being A2, very, very simple, one HLA molecule, a very, very simple type. As of last week, there were over 1,400 different subtypes of A2 alone that we have to be able to differentiate at the point of transplantation. So this is a huge challenge for us in the HLA typing field and in our clinical laboratories. One of the ways that we've chosen to combat that is by using a technique called third generation sequencing. And this was implemented into our clinical labs a few years ago, and we are using it in our research projects going forward. And what this technology has allowed us to do is to sequence through the whole gene in one go. And what that's allowed us to do is gain more information about the gene than we've ever been able to do before. And we're able to do that in a time frame that's much quicker than previously. So we can do, in a couple of hours, we can get information that historically could have taken us a couple of months. So this has really changed what we're able to do and the information that we're able to gather. But does this additional information matter? We're getting more information than we've ever had before. We're understanding more about HLA. Does it actually matter for the patient? Does it result in different outcomes? And that was one of the first questions that we wanted to ask or to answer once we had implemented this new technology. And so we took the patient donor project cohort of patient and donor pairs, and we went back and typed them using our third generation sequencing or TGS technology. These individuals had been transplanted between 1996 and 2011 at a UK transplant centre, and it was a mixed cohort of adult and children, but they all had a blood cancer. So this is a very busy slide, and don't worry, I'm going to talk you through it. But what we have on the left-hand side here is the previous match. So this is how well matched we thought they were at the time they were transplanted. And then along the top here is showing how well matched they were after we had gone back and done sequencing using our TGS system, our third generation system. And so what you can see here is that those individuals that were a 12 out of 12 match, so that's matched for both copies of the 6 HLA genes that I mentioned that we believe we should be considering for matching patients and donors. Of those 12 out of 12 matched individuals at the point of transplant, when we went back and recharacterized them, we actually found that only about 75% of them remained that well matched, with the remaining pairs becoming less well matched, all the way down to a 9 out of 12, meaning there were 3 mismatches that we couldn't identify using the techniques that we had available to us at the time these individuals were transplanted. And so on and so on, and that's what this information in this table is showing. But one thing that was really interesting to us was that the more known mismatches that we had at the time of transplant, the more unknown mismatches that were also present. So you can see here that, as I said, it's about 75% of those individuals that were a 12 out of 12 match at the time of transplant remained a 12 out of 12. And that was the same for those that were 11 out of 12. But as we had more known mismatches here, so 10 out of 12 and 9 out of 12, we saw a less percentage of individuals remaining that well matched. And if we took 9 out of 12, for example, here, just over half of them actually remained a 9 out of 12, with people even going down to being a 5 out of 12. So this technology really is highlighting differences that we just couldn't characterize at the time that these individuals were transplanted. So in terms of patient outcome, one of the first things that we wanted to do was to say that these individuals that I've highlighted at the top here, those individuals had a 12 out of 12 match. So they should have done the best in terms of outcome. But if we now have a look at these individuals and we compare those who remained a 12 out of 12, and we compared their outcome to those who actually we found were not as well matched as we thought, do we see a difference in how their outcome has been after their transplant? And what we can see here is that there is a significant difference in their overall survival. So what we have here in the blue line at the top, these are individuals that were matched at a TGS level. So after we've gone back and recharacterized them, they remain a 12 out of 12. And green line here at the bottom are those individuals that we thought were a 12 out of 12, but actually after this extended typing, we find that they had mismatches that we didn't know about. And you can see that the 5-year difference in overall survival here is almost 25%. So this is a huge difference in outcome for these particular patients. What we also did was take those individuals where we thought that we were providing them with the best possible donor at the point of transplant, which at that point was a 10 out of 10 match. And we stratified them as to whether they were matched for this 6th gene, which is known as DPB1, in various different ways. And we also combined that with CMV matching. So CMV stands for cytomegalovirus. It's a common virus that many of us will encounter in our lifetime, will probably have a few quite mild symptoms and will recover. If you're a healthy person, if you don't have any underlying health conditions, you'll recover from CMV, but you will remain CMV positive. You will have some dormant virus in your body that your immune system will control for the rest of your life. However, if you are immunocompromised, for example, if you're a patient undergoing a stem cell transplant, CMV becomes a much bigger deal, and you need to be able to control CMV in these patients who are challenged with a poor immune system. And so what we've done here is to stratify out how not only HLA typing for the 6 genes, but also matching for whether the patient and donor have ever been exposed to CMV before. And what we can see here is that there is almost a 40% difference here in survival based on those individuals that we thought we had given them the best possible match, a 10 out of 10 match. Actually using this additional information and including matching for CMV, we can actually stratify that outcome hugely. And so we're knowing now what is the best possible donor that we can find for patients based not only on HLA, but including factors like CMV as well. So I've shown you that matching for HLA is very important, but for some people, we don't have a choice. They don't have an option of a perfectly matched donor. And so one of the questions that we've asked is, are mismatches equal? Can we pick any mismatched donor and this patient will have the best possible outcome that they can have? Or are there certain mismatches that we should be avoiding? And what we've actually found is that there are some mismatches that are worse than others. So this is again data that's come out of the Patient Donor Project. And what we've done here is look at those individuals that have a 10 out of 10 match, so the best possible option that they have. And we've compared those that are mismatched between those that were mismatched at a 9 out of 10 levels. There's a single mismatch here, but for one gene known as HLA-B, these individuals were matched. And the green line at the bottom here are those with a single mismatch, again, a 9 out of 10 match, but their HLA-B gene was mismatched. That's where the mismatch was. And you can see that if you have an HLA-B mismatch, your survival is significantly worse after the transplant. Going back to the 6th gene that I was talking about, HLA-DPB1, we also know that this gene is possibly a gene that we can consider mismatching for. And actually, what people in the community have been working on, and Anthony Nolan has contributed to this data, is that there are ways of determining permissive mismatching at the DP molecule. And what that means is that we can mismatch at DP if we have to, and that actually there's no significant detriment to the patient in terms of their outcome. So this is data that actually came out of the 3rd generation sequencing study, and this mirrors what other people have found previously, that while having a match is the best possible option, actually having a DP permissive mismatch, but you're matched everywhere else, doesn't result in a significant difference in outcome. But you want to avoid the non-permissive mismatch in this particular example, for obvious reasons. The non-permissive nature of this mismatch is resulting in worse outcomes for patients. So we're able to look at mismatching now, or beginning to be able to look at mismatching in this way and say, which mismatches can we tolerate and which ones can we not? So moving on a little bit from HLA, what other clinical factors do we have that are impacting on the transplant outcome? One of the things that we do know is that donor age can have a significant impact on transplant outcome, hence why our recruitment and our joining age for our register has been reduced in the last 5 or 6 years. And what you can see here is that survival for patients is significantly higher when a donor less than 30 is chosen in comparison to when a donor over 30 is chosen, and that's the green line at the bottom here. And this is data that's been mirrored in other studies. This was a big international study that came out of the USA. And again, you can see those patients receiving a transplant from a donor between the ages of 18 and 32 in this instance, but their survival was significantly higher than the other age groups. And the reason for that, so this graph actually just shows here, the purple line is those donors that fall into the 18 to 32 category. And on the left-hand side here, we have the impact on outcome of using donors in the 33 to 50 age group and then greater than 50. And you can see that the consequence of using an older donor, the problems that arise are due to conditions such as GVHD, which does have an impact then on their mortality. So we are actually selecting younger donors, transplant centres select younger donors, and we are recruiting younger donors because it gives patients the best possible chance of a good outcome after their transplant. I've already mentioned about CMV in the earlier study, but this is just to show again that matching whether a patient and donor have ever been exposed to the CMV virus before does impact on the survival. So again, if you're having patients and donors that are matched as to whether they've ever seen it before, their survival is significantly higher than those who are mismatched. And following on from the earlier analysis, you can see here that the HLA matching and CMV matching, it gives the best possible outcome. But if we can't find a CMV matched donor, a CMV mismatched donor will reduce your chance of overall survival, but it's not as detrimental as having an HLA mismatch here. With a CMV match, and then the worst possible option here is an HLA mismatch and a CMV mismatch as well. And I'll just briefly touch on some of the non-HLA genetic factors. As I said, this is an area that we're really just beginning to get into in the laboratory and try to understand some of these additional factors. And these are a few pieces of data that we've published from the Patient Donor Project over the last 10 to 15 years. In fact, this study on the right-hand side has just been published earlier this year. And these are genes, the left-hand side here is looking at a gene called NOD2, and the right-hand study is looking at combinations of KIR genes, which are the killer cell immunoglobulin-like receptors. These are genes that have never been considered in terms of transplantation before and are not routinely matched for in transplant practice. But what we can see here on the left is that this is specifically here for patients with acute leukemia. But what we can see is that if you have the wild type or the normal form of the NOD2 gene, patients have a significantly higher chance of having a positive outcome in their transplant as compared to those who have a mutated form of this NOD2 gene. So this is an additional way, if we could look at adding in this information into donor selection, we might be able to help improve transplant outcome using this additional factor. And here again, this is looking at the KIR genes just on the right-hand side here. And what we can see is that certain combinations of KIR genes in the donor result in differences in overall survival here. And again, these are not markers that we're routinely using, but these are things that we can be considering to add into donor selection going forward. So just to summarize really, the Anthony Nolan Patient Donor Project really has changed practice, and that's not only in the UK or at Anthony Nolan, but this is internationally as well. One of the really important things about the Patient Donor Project and some of the work that comes out of our research institute is that we translate this into practice as soon as we can. So we are able to put things into practice in our clinical laboratories. We are able to factor things into our donor selections. Our search team are changing how they're searching for donors and who are being selected for matches. And we do that as quickly and as safely as we possibly can. An example of this is that the DPB1 studies, we've actually, we've been doing a lot of work on DP over the last 20 years that I've worked at Anthony Nolan, and we have been routinely including DPB1 into our donor typing since 2013. And that's really led the field. That was a lot of other registries have recently been adding DP into their routine typing. So we really led the field on that one. As I've mentioned to you about donor age, we and others have shown how important donor age is, but Anthony Nolan has reduced our minimum age to join the register. We let people join the register from the age of 16 to 30. And that's as a direct response to knowing that this is the best possible donor that we can find for our patients. And not only that, haven't just changed our recruitment ages, but we're actively trying to recruit younger donors to help patients. So our average age of joining the register at the moment is 22, and the most common age is 17. We're also able to recommend what kind of matches patients should be finding for their donors. And 12 out of 12 matches now are becoming the norm. That's what people are striving to find for their patients. But if they can't find that, then we're also able to help them identify what mismatches they should be avoiding. For example, looking for HLA mismatched donors and CMV mismatched donors, we should avoid that at all costs. And I would just like to thank all of the transplant centres. This is all of the centres individually here, because we couldn't do this work without them. And as I said at the beginning, we couldn't do this work without all of you as well. And we are very grateful for the support that you give us that allow research such as this to take place. So thank you very much. And I will take some questions if there are any. See if I can.
Thank you so much. That was like super informative and so interesting to sort of see the reasoning behind sort of like why we do have our targeted recruitment and how important that all is. Yeah, thank you so much for that. We've got a couple of questions coming in now. So
we do. Yeah, so somebody has asked, they're wondering what role bioinformatics and AI have on the future of HLA matching and the stem cell register. And that's a really great question, actually. And I wish I'd had half a day to talk to you about all of the research that we do at Anthony Nolan, but we do have a bioinformatics team who part of their role is working on understanding the register. So some of the type of work that they have done is looking at the breakdown of tissue types across the UK. And actually breaking that down into regional variation. And what we can then actually do is have a look at our register and see when we don't have certain proportions of certain types of donors and certain matches, we can actually know where in the country we can go to recruit from that actually allows us to have a greater chance of identifying donors with these particular tissue types. So it's actually looking at the breakdown of the UK and helping us to guide our donor recruitment using bioinformatic tools to be able to do that. Some of the other ways that we're using bioinformatics is to look at who is on our register and look at the breakdown of different phenotypes within our register, the common phenotypes that we're seeing, and which are some of the non-common phenotypes. And when I talk about a phenotype, what I mean is the combined information of a tissue type of an individual. So it's the two haplotypes, the two bits of information that we inherit from our mum and our dad. And we're looking at that, and what we're actively doing is going to those, we find those individuals, we're looking to see who are the most common phenotypes in our register, and we're actively getting back in contact with those individuals. We know that their phenotypes are common. We know that that means that they're probably going to be chosen because patients will have the same phenotypes as the donors, so they're more likely to be chosen. And we're making contact with those individuals, we're characterizing them to the best possible level that we can as part of our phenotype project. It's essentially what we're trying to do is enrich this panel of super donors so that they are primed and ready to go for patients as quickly as we possibly can, should they be needed for transplantation. So bioinformatics is playing a huge role in the work that we do. That's just a small snippet, sort of window into some of the work that they're doing. But it definitely is shaping registers, not only at Anthony Nolan, but across the world, and is really going to be the future, I think, for bioinformatics. Somebody has asked, does money that Marrow Groups and Anthony Nolan raise go towards helping with research funding, or mainly to patient services and adding donors to the register? The money that Anthony Nolan raises, it goes to a wide variety of different things, including some of the research that we do. It's a proportional breakdown of the funds that are raised, but some of the funding will go towards some of the research that we do.
I think also just to add into that, like for Marrow, the reason we talk about sort of like how it's £40 to add someone to the register is for a fundraising, from a fundraising perspective on the ground, that's a really tangible, easy to understand concept, and that's like a small enough amount to encourage people to donate, like a small, like a pound, a couple pounds. So that's why maybe the Marrow fundraising strategy sort of focus more on adding people to the register, but like Neva said, really it just goes into a big pool which is just spread evenly.
Yeah, and it frees up other money that allows us to carry on with our research. Somebody has asked whether the rate of relapse is affected by matching. We see that sometimes in certain cases certain genes, so DPB1 is quite an interesting gene, and what we have seen is that sometimes mismatching for that gene actually results in lower rates of disease relapse, so it's actually protective. And one of the reasons that we think that that might be the case is that your immune system needs a little bit of genetic disparity between the patient and the donor to kickstart lots of different immune responses, but one of them is the graft versus leukemia effect. So it's the cells that are coming in from the donor, we want them to be stimulated to attack any leukemic cells or any disease cells that might be residual in the patient. And so a little bit of genetic disparity, and we think DPB1 is an example of a gene where mismatching actually allows that to happen. Just, it's a little bit of an immune response, but it's focused in the right way. So matching does help with relapse in a very unusual way, but in an exciting and interesting way too.
So I think we've just got another question in the chat. Chloe says, hypothetical situation, if someone has a 12 out of 12 HLA match with a donor but a CMV mismatch, is this better than having an 11 out of 12 HLA match but with a CMV match?
So the data that we have shown, I will try, and I don't know if I can go backwards with my slides. I will try and just cover this really quickly. This is the data that has come out of our TGS study. And what you can see here is that the line at the top here is a 12 out of 12 CMV match. And we are looking at 10 out of 10, but permissively mismatching for DP in this instance. And you can see here that the next possible best option is a 10 out of 10 permissively DP mismatched CMV match. So actually, in order of outcome in this particular data, you would be better off picking a reduced mismatch or a permissive DP mismatch, but a CMV match over a 12 out of 12 but CMV mismatched individual. The one thing to point out is that although the risk here, it looks quite different, statistically, there is no difference in outcome between these first 4 lines just here. So any of the individuals that fall into the 12 out of 12 or 10 out of 10 permissively DP mismatched individuals, regardless of CMV matching, had, there was no statistically significant difference in their outcome. But in practice, you would probably go for the higher HLA match, and certainly based on other data that we've had as well.
Okay. So we don't have any more questions at the moment. Maybe we'll give another few seconds just to see if anyone else has got any.
It's too much science for Thursday.
I think we've already had some really great questions, actually.
They've been really great questions.
Yeah, otherwise, I would say probably that's the end of the Q&A. Don't think we're going to get any more questions. Oh, we're getting a couple of thank yous in the chat. But yeah, do you have any final comments, I suppose, Nima?
No, just to reiterate what I said, just thank you very much. I hope this has been not only interesting for you, but maybe helped understand why we're working with you and helping to recruit the kind of donors that we're looking for. And thank you very much.
Yeah, I suppose a final thank you to your team and all the amazing work that you guys have done to get us to this point in the first place. So I'm going to stop recording now.